the no audio. We didn't realize that. So we can start. I'm sure they'll come in sooner or later. Like always, make sure to go sign in. We'll post the description, or we'll post the link to sign in the comments. So make sure to sign in. And what I was saying before that you guys couldn't hear was that to start off with mitosis and meiosis, we can go off. We're going to start off with a little bit of background, just really quickly, because the chromosomes that we work with are a type of DNA packaging, basically the ultimate type of packaging. So what I was going to talk about was packaging. So the way you can kind of remember, so there's four levels of them. 4 levels of packaging. The first one is going to be 10 nanometers. That's when you just have a histone core with DNA wrapped around it. That's what this ugly thing is. Yeah, that's what that ball is. I remember that ball, that histone core is made out of 8 different histones. Just 2 of the H2s and 3 of the H4s, those, like the 2As, 2Bs, H3 and H4. And remember, it's the histone core does not include the histone 1 and the histone 1s. The way you can kind of remember that is that when it comes to like having siblings, if you're the oldest, right, you're the firstborn, you're the one that helps kind of parent the other ones. So in a way, if you know what histone 1 is, or if you know the purpose of histone 1, which is to help this process where you have the DNA wrap around the histone core, you can kind of think of histone 1 as like the firstborn that's helping its siblings out by wrapping around the DNA. That's the way I kind of remember it. Because you do need to know which ones are in the core and which ones aren't in the core. Yeah. So moving on from that, once we have the histone core wrapped with DNA, now we can have other histone cores wrapped with DNA, right? And that's where we get into more condensed. Now we're doing 300 nanometers. Yeah. And so once we have enough of those histone cores wrapped with DNA next to each other, they start condensing on each other. They start interacting, they start condensing onto each other. That's when we're working with the 300 nanometer domain. And then once we have enough of that condensing, they start forming these things called loop domains. So basically, they look something like this. Okay, and so that's what a loop domain is. However, because these loop domains, they're not as stable as you would think. They need to have like a base to kind of grow on. So what they do is these loop domains at the 300 nanometers level, they attach themselves to proteins. So that's what you have at the 300 nanometer level. And so once you have the loop domains on scaffold proteins, eventually you'll get to the 700 when you have more than plenty of that scaffold protein in loop domains. And so that's what we see. So once you have enough of those, you start seeing this kind of formation where you have two scaffold proteins with loop domains on them. And if you guys can kind of zoom out from that picture, you can kind of tell that it's a chromosome. And so that's what I meant by the chromosome being like the ultimate type of packaging. And so, yeah, that's all you need to know about DNA packaging. And yeah, now we can move on to meiosis and mitosis. Unless you guys have any questions about these, just put them in the, if you have any questions at all, just put it in the comments. But right now I'm going to go into the purpose of meiosis and mitosis and just a general overview. Huh?
Okay, that's it. Pretty cool. What's that called? Huh? It's better than pretty cool. It's better than that.
Okay, so when we think about meiosis and mitosis, the best way to think about it, or the purpose behind them, is that meiosis produces. Oh, somebody has a question.
So the 10 nanometer one is just a histone?
Yes, yeah. So going back to the DNA packaging, yeah, that's just this is the 10 nanometer packaging. That's when you just have one core with the linker DNA wrapped around it. Remember, and then in the core it's just H2A, H2B, H3, and H4, and then H1s are on the outside of the histone core. Yeah. So when it comes to meiosis and mitosis, it's good to know what the purpose of them are, because then you realize it makes sense. Or if you know what the purpose of meiosis and mitosis is, then it helps you remember the steps of them. So for meiosis, the purpose of meiosis is to create gametes. And gametes, like I drew up here, can either be sperm or egg cells. So the way you can kind of think of it is that, and remember, at the end of meiosis, you have different types of cells. And so in meiosis, you have 4 different types of cells, right? And which makes sense, because if you look at your brothers and sisters, you don't all look alike. And the reason is because meiosis produces these gametes that are all different from each other. And so that's why this sperm cell might be similar or might be completely different in genetic material than your brother and sister, because meiosis produces those 4 different types of cells, which makes sense because you don't want your brothers and sisters to be clones of you. And that's not what we see in actual life. But mitosis comes in once a sperm and egg cell come together and form a zygote, and that's a single cell, a single nucleus. Now, for that single cell to become, eventually you or your brother or sister, they have to go through mitosis, which makes sense. What's that say? Okay. Which makes sense because the way you can kind of think of it is that mitosis is for somatic cells. Which makes sense because for somatic cells, that somatic cells is just a fancy word for cells that are anything but sperm and egg. So they could be kidney cells, they could be liver cells, they could be brain cells, anything like that. And when you think about it, when you have, let's say you have a cell right here, a skin cell, right? If you, let's say you have a cut and let's say some of your skin cells die, you still have some other remaining skin cells, hopefully. Depending on how big the wound was, but whatever. Those other cells, they'll start mitosis and they'll form new cells to make up for the ones that died. Now, if those cells went through meiosis, then you would start growing eggs and sperm instead of the exact same kind of cells that you want, which are skin cells. So you want the cells that are coming out or that are being produced, the skin cells, you want them to be exactly the same. That way you can recover whatever cells that died off. And also that makes sense when you're talking about, in the case of your liver, if some liver cells die in your liver, you don't want new cells that have new genetic material. No, you want the identical types of cells that you lost, because you're trying to make up for that loss in your liver or in your heart, anywhere in your body, besides making sperm and gametes, or just gametes. Yeah. Does that make sense to everybody? If you guys have any questions about this, make sure to post it in the comments. Also, make sure to sign in. Just take a quick second, sign in, come back. And then if nobody has any questions about this, I will go into PMAT, which are the steps of mitosis and meiosis, which I'm sure you all came for. That's the little screen over here. You're good. So, pretty good pictures. Okay. It's only covered in time. It should be fine. So, we're going to go one step at a time. So I just drew the first picture. So PMAT, if you guys don't know, stands for prophase, metaphase, anaphase, telophase. In this example, I kind of include, I'll go into cytokinesis when we get to it after telophase. But PMAT is just a way to remember the names of the steps, or the order and the names of the mitosis and meiosis cycle. So in the very beginning, right here, when we have this cell, this right here is we're in the G2. If you guys remember the cell cycle, G2 is the the part of the cycle that comes right before we go into mitosis. And right now, when we go from G2, if you guys look, it's all shaded in. What that means is that in G2, you have all this DNA kind of like loose and kind of dispersed everywhere. It's not very condensed. It's just all around the nucleus, which makes sense because you don't want. It makes sense because you're not moving around the DNA. You're not doing anything to it. In S phase, you're maybe replicating it, but you're not moving anything. You're not doing anything to it. Now, in prophase, that's when you're going to start seeing the chromosomes right here, that DNA, start packaging into DNA chromosomes. Why? Well, because for the rest of this phase, the way Derek kind of put it, which is a good analogy, is that when you have a slinky, per se, when you have a slinky, let's say you extend it across the room, right? If you aren't doing anything to the slinky, if you're not moving it around or anything, you can play around with it. You can just, move around with it when it's all stretched out, right? But now, if you want to take it somewhere else, if you want to move it at all, well, there's a chance that they might start twirling around, or it might get, it might just get into the weird conformations and stuff. And so you don't want to move it around when it's spread out everywhere, because it can tie against each other and all that stuff. Now, so that's basically what's going on in G2. When you're going to prophase, you're like, that's the step that signals, hey, we're going to go through mitosis. So let's start packaging that DNA or start condensing that slinky. That way we can play around, with it and don't have to worry about anything breaking or getting tied up or anything like that. And so right now we're in the phase where the slinky is kind of stretched, or not stretched, back to the regular shape. And so what these things are right here are called centrioles, and those are going to be very important when it comes to for this whole process. And what they do is they create what's the name for it again? Centrioles. What do they create again? Mitotic spindles. That's the name. Mitotic spindles, which are these little leg things that you see growing out of them. Those are going to be important once we go into metaphase and then anaphase and the rest of the cycle. And you'll see why. But the big thing about prophase is that the DNA starts condensing. The nucleus that surrounds this, the nuclear membrane that surrounds that nucleus, it starts breaking apart because we're about to start the cycle. We're going to start moving that DNA around and all that stuff, all that good stuff. So now let's go. If anybody has any questions about that, put in the comments, but I'm going to move on to metaphase now. Yep. Yeah. Right. So when it comes to, maybe I should move this closer. Okay. Cool. So when it comes to metaphase, the way you can kind of remember metaphase is meta, or it starts with an M, right? So middle, meta. And the reason why that's important is because there's a thing, or the chromosomes, once they get out of this conformation, out of their nucleus, and the nucleus disappears, they align to, they get pushed to the middle of the cell. which is called the metaphase plate. It's not a physical thing. You won't find it if you dissect a cell, or you won't be able to see it. It's just signifies the middle where all the chromosomes kind of line up. So what happens is the spindle fibers from these centrioles start growing and growing and growing, and eventually they'll start connecting to either of the sister chromatids of a chromosome to the centromeres, which is the middle part of a chromosome. And then they'll do that for each one. We're working with 3 right now, 3 chromosomes, and they'll attach to each one. And so that's what I wrote right here. Yeah. And then after that, we're going to go to anaphase. And the way you can kind of remember that is it starts with an A, so you can think of away, right? Because in anaphase, you have a splitting of them, or you have the splitting of the chromosomes. So what you guys can see is that in anaphase, you start seeing these mitotic spindles that once attach to the centromeres, right here in this step. Once they attach to those, they start retracting, they start growing, they start shrinking. And so what that does is that it forces the chromosomes to split into their sister chromatids, or it breaks apart the sister chromatids, which is the confusing part because for this right here, for this step, we have 3 chromosomes. Oh no, I'll get to it at the very end. So when we had that chromosome, we split it into it was the chromosome with the sister chromatids. We split that, right? In anaphase, and the way you can kind of remember that is ana away, away, meta, middle. I don't have anything clever for prophase. Yeah, but yeah. So then you have the split by the mitotic spindles shrinking back, and so that's why you have this. That's where the arrows are pointing. They start going back, and now once we move on to telophase. Now we move on. Once we move on to telophase, we have telophase. We start seeing the mitotic spindles keep growing and growing and growing, therefore pushing on the side of the cell. And once they push enough on the sides, of the cell, you start seeing the cell have like a cleavage form, and it's called the cleavage furrow. Once they push enough, the cell will become kind of almost 2 cells, but it's not there yet. And so once that happens, once that pushes enough, it'll still keep on growing, and then eventually you'll go through cytokinesis, in where you actually have the splitting of the cells into 2 identical daughter cells in the case of mitosis. And so after this, we're going to see, I'll draw on the board. So after cytokinesis, which is basically just a word that signifies the splitting of a cell into two cells, you have these two daughter cells. And the thing about mitosis, like I said, the purpose of it is to create identical cells, right? So you create from this one cell, you create two identical daughter cells. And the way you can kind of, the way that, sorry. Three was a bad number, but so the way you can kind of remember, or at the very end of mitosis, once we go through all these three steps, you'll have two identical daughter cells, and so both two identical daughter cells that both have 2n equals 3, 2n equals 3. 3 was a bad number because then you imply that n would be 1.5. Just think of it as 4. So 2n equals 4 and just add another chromosome. But basically, the important part is that you get two identical daughter cells after mitosis, after this whole process. And even though I said I'll talk about misconceptions at the end, I just want to throw out that the reason why people get this confused is because you go from this chromosome that has two sister chromatids into this chromosome at the very end, and you still call that a chromosome, which is confusing, but at the same time, in a way, makes sense because this chromosome, this sister chromosome, when you have that chromosome that's made out of two sister chromatids, they are the exact same copies of each other. Because of through the S phase, you duplicated one and got to the other, or this would be a better example. You duplicated one to the other, and so they're basically clones of each other. And so once you split those, even though they're clones, they're now individual chromosomes. This is one chromosome, where this is a sister chromatid, or this is a sister chromatid. But once you split them, they're considered chromosomes by themselves. And so that's why we still retain that 2n equals 3 at this end. And we go from 2n equals 3 to 2n equals 3 on this one, even though this one, there's 3 chromosomes, and they have six sister chromatids. And in this one, there is no sister chromatids. And that's why we're able to keep the 2n equals 3, because once you split those chromosomes into sister chromatids, or you split the sister chromatids of a chromosome, it becomes one chromosome and another chromosome, which is kind of confusing. It took me a while to understand that. But if you understand that, then it makes sense why you're able to keep the 2n equals 3 to 2n equals 3. Yeah, so I'll let you guys digest that or process that for like a minute. Put any questions do you guys have about this whole process, or if you have any questions about anything else, I'll give you guys a minute, and and then after this we'll go through PMAT, but we'll go through PMAT of mitosis or meiosis 1 and meiosis 2. So, what I just realized is that although we just went through this, there's a lot of terminology that we that I just kind of assumed that you guys know. So let's go back a little bit and talk about some of the terminology. So I'm going to erase this. If you guys want to take a picture of it or take a screenshot, go ahead. Okay, cool. Or also you could just rewind the video. But we're going to go through like terminology of chromosomes, maybe what a diploid haploid is, what a chromosome is, what a sister chromatid is, when is it diploid versus haploid, stuff like that. We're going to go through that right now, just so you guys get more familiar with the terminology, because in these processes, we go through them we just kind of throw them out there all the time. And so it's important to know what we're talking about. So I'm going to go through that right now. Is this? Oh, nice. Is it muted? No. Muted. All right, so the type of terminology. Oh man, there's a glare. All right, at least there you can see the terms. So when it comes to the terminology behind replication, or not replication, chromosomes, the big words that we kind of throw out, at least right now for the first four, is haploid, diploid, somatic cells, and gamete cells, or Germanic. So basically, what we say when we say haploid, that means one set of chromosomes. Now, what does that look like? So, when I'm talking about haploid, that means one set of chromosomes. The way you can kind of remember that is that it's not diploid, where diploid is diploid, di meaning two, like dimer, what other things? Yeah, mer meaning two. So haploid is just one set. So what does that mean? That means that the most like relevant example most of us would think of would be sperm cells and egg cells. Like we talked about in class, sperm cells, that only has the father's DNA in it, and that's one set. That's one set of these 23 different chromosomes. Now, the egg cell, that only has one set of the mother's DNA chromosomes. So each one are considered haploid cells. Once these two come together and they form a fertilized zygote, So once they come together and form this fertilized zygote, I'll write that down. Fertilized zygote. Once they come together and form the fertilized zygote, that's considered a diploid. Why? Well, if haploid is one set of chromosomes and you have two of those one sets come together and form a zygote, now you consider it a diploid because now you have two sets of chromosomes. And so the nucleus of the first of that one cell of the fertilized zygote, that's going to contain, it's going to be, instead of being n equals 23 chromosomes for the egg cell and n equals sperm, or n equals 23 chromosomes for the sperm cell, once those come together, it becomes a diploid. And we notate that with a 2n because n represents, n number of sets. So n just represents the number of sets. And so when n equals 23, n represents numbers in set. Sorry about that. N just represents the number of chromosomes in a set. So when I say n equals 23, that means that in one set, there's going to be 23 chromosomes. That's why haploids have one N. When we're diploid, once we have those two come together, we notate it by doing 2n, which means that we have two sets where n equals 23, and so 2 times 23 gives you 46, which means that you have a total of 46 chromosomes, but they're separated into two different sets, where one is from the mom, one is from the dad. And that's what I'm trying to show here. 2n equals 46 because n was equal to 23, which means the number of chromosomes in a set, and 2n is the total number in a diploid.
You might want to say that 2n carries information from both parents.
Yeah. So, like Jorge pointed out, 2n means that it has one set from the mom and one set from the dad. That's what I've notated right here. And so that means that it carries information from both your mom and your dad to form that one fertilized zygote. Now, from here, like I mentioned in the beginning of the stream, you can go through mitosis. The cell can go through mitosis to start forming all these other different types of cells, like skin cells and all that, organ cells and all those types of cells that you might need to eventually grow into an adult or a fetus in this case. So now, this kind of transitions us to the next two terminologies that we're going to go through.
Do you want to put like an example of just draw n equals 5 versus, or n equals 10 versus 2n equals 10?
That's a good example. Yeah. So, n equals 5 versus.
Or just n equals 10 versus 2n equals. Or just to make it easier to draw, n equals 4 versus 2n equals 4.
That's fine. So n equals 4. There's no other color though. It's not that great. Oh, I could try it. Let's see. No, that's right. That's right.
Just say something like, oh wait, no, you need to extra this 2n equals 8. Those two are the same chromosome, but then one is from the dad, one is from the homologous.
Let's do. No, that was right.
So these two carry the same information. One's from mom, one's from dad, and all of this ones carry different information.
Yeah. So as Derek, or not Derek, sorry. Yeah, go ahead.
Just say something like this is mom, dad. And then let's just call it chromosome. Or we could just call it chromosome number, let's just say 1, and then chromosome. Seven.
Yeah. So, yeah, so like how Jorge drew out, it's a very, very good thing to know the difference between these, right? So n equals 4, what does that mean? Well, that means that, like I said before, over here, n is the number of chromosomes in a set, right? So if n equals 4, that means you have 4 different chromosomes that are contained within one set. Now, if you were looking at 2n equals 4, well, that's different because now you're talking about, let's say, just really quick math. 2n equals 4, well, that means that n equals 2. What does that mean? Well, that means there's 2 chromosomes in one set. What does that mean? Well, for, let's say, what does that n equals 2 come in? Well, that's 1 from the dad, or when it comes to the dad, you're going to get 2 from the dad, and then 2 from the mom to form that 2n equals 4. And the way you can kind of distinguish these is that for 2n equals 4, there's going to be 2 chromosomes that are kind of similar, or similar enough that they're considered homologous, or rather, another word for it is homologs. It means that it just carries the same kind of genetic material. So basically, when it comes to what Jorge wrote, that first set right there, the first 2 homologs, those 2 carry basically the same genetic material. And so those, let's say that's chromosome 1. This is chromosome 1, that's chromosome 1. They kind of attract to each other because they carry the same kind of genetic materials, while these other 2 are considered homologous because they also carry the same genetic material to each other. And one is from the mom and one is from the dad. And so once they come together, they kind of pair up. And so that's basically the difference between n equals 4 and n equals 2, 2n equals 2, or 2n equals 4. If you guys are just joining us right now, I see a couple more new people, make sure to sign in. And also, if you guys want to see what I was talking about before, you can kind of rewind the video a little bit back to kind of catch up. But also, yeah, so that's the difference between n equals 4 and 2n equals 4, which is very important, especially for this exam, because I'm sure you're going to have plenty of questions about that. Anything else to add?
Talk about a karyotype.
Karyotype?
Yeah, like give the difference between 2n equals 46, which are somatic cells, versus n equals 23, which are sex cells. You know what I'm saying?
N equals 23 is. Oh, okay, gotcha.
If you want, I can explain.
Yeah.
You can take a break if you want. It's up to you.
Sure, if you want to go through. Yeah. Oh, did you need that up? Did you need
No,
that?
this one.
All right. So I'm going to take a quick break, and Jorge's going to sub in for me, and he's going to explain
Yeah, somatic cells.
somatic cells and other stuff.
All right. Hello, everyone. So just, can you add a picture in here?
Add a picture?
Yeah, like, can you, no, I don't think so. So we're just going to go over, like, because you need to know what karyotypes are. Is that what it's called, karyotype?
I think there's only one Y.
Karyotype.
Well, no, karyotype. Yeah, I think that's.
Okay. So all living organisms have a specific karyotype. I'm pretty sure you guys have heard before. Can you see that? Yeah. Well, humans have karyotype 2n equals 46, right? So that means that we're gonna have pairs of chromosomes. We're gonna have 23 pairs of chromosomes, right? So the total is gonna be 46 chromosomes divided in 23 pairs. Each pair comes from one of the parents. So like For example, just like we did before, we divide the chromosomes sets like 2, 3, 4, 5, 6. If we did just an example, 2n equals, let's say 2n equals 12. That means that there are 6 pairs of chromosomes to give a total of 12 chromosomes. And for example, let's just say the first set will be, let's just draw this one, and it's conjugated or just complementary, not complementary, just homologous to this draw the same size. And one of this comes from mom, one of this comes from dad. And then your second set would be, let's make it a little shorter, homologous to another one. This is from mom, this is from dad. You can keep going like that. And if you look at the picture for humans, you have 23 pairs to give a total of 46 chromosomes. Remember that one of them is coming from mom and one of them is coming from dad. That's just your somatic cells. Remember that somatic cells are they have information with both parents. So somatic cells are called 2n, whereas sex cells carry only information from one parent. So these are just n. So this kind of cells you're gonna find in pretty much just everywhere in your body. Like all your organs are gonna have this somatic cells, whereas this ones, of course, they're gonna be your like reproductive organs, just your sex organs, where you divide this into eggs and sperms. So this sex cells are the ones that join together to give you your somatic cells. So I'm just gonna erase this. So again, somatic cells, you see that, are pairs. These are pairs coming from both mom and dad, right? So if I have an example of 2n equals 4, those are going to be, this is from mom, this is from dad, this is set number 1, and this is set number 2, or pair number 2, pair number 1. And remember that this one's carry the same information. This one carries the same information as this one, but this one has the information coming from your mom and the information coming from your dad is here. So these are found, let's just say, you can find these cells like in your liver. You're gonna see, if you look at the karyotype, like if you pull like a sample from your liver and you look at just pretty much the genetic material in it, you're gonna see somatic cells which carry like information for both your parents. Whereas if you go, you can just see liver, your eyes, I don't know, your heart, you're gonna see all these kinds of cells in there. Whereas if you go to any reproductive organ, is that how you call it?
Mm-hmm.
Sex organs.
Yeah.
You're just gonna see sex cells. So in case of the female, you're gonna see eggs, and for male, you're gonna see sperm. And remember that for this one, it's only gonna carry just the information from what is just, I'm gonna call it the female information. And this one's gonna carry, of course, the male information. So if you look at a sex cell that has a genetic material n equals 4, that means that we're gonna have 4 sets, and they're all gonna be different, but they're all coming from, let's say this is a sperm.
4 chromosomes.
Yeah, it's gonna have 4 chromosomes. And they're all different. They all carry different information. Let's just call this chromosome number 1, chromosome number 2, chromosome number 3, and chromosome number 4.
But they're all from the same.
They're all coming from the same parent per se, which is called, if it's a sex cell, it's gonna come from that. And if it's an egg cell, it's just gonna come from female, right? So if the key here is to like distinguish it, like just the difference between somatic cells and sex cells. And if you're given a karyotype of an organism, if you see that they're split into pairs, or they're homologous pairs, then you're supposed to know that, okay, this pair, there's pairs here, that means that one of the chromosomes comes from mom, the other chromosome comes from dad. So these are somatic cells, and you're supposed to just divide by pairs. So like I said before, 2n, this means that it's coming from both parents, that means that it's divided in homologous pairs. We're going to have a total of 46 chromosomes for humans. That means that there's 23 pairs. Whereas if you have n equals 23, those are your sex cells. And it's only information from one of the parents. Only one parent. So yeah. Does it look good?
I can see it.
Just try to read that, and if you have any questions, just feel free to ask, because this is very important you understand this. Because if you have to understand this, you can move on to mitosis and meiosis. What else can you say? For example, just an application of mitosis. This has to do with. Can I erase this? Are you good?
You're right.
So the way I like to think of mitosis, I relate it to just organism growth. So as you're growing up, of course, you're like increasing in size and like so on. You just think of it like your organs are just increasing. That means that you're just duplicating the same information. And like I said before, your organs carry somatic cells. So whenever you do mitosis of somatic cells, like you're just making the same, you just like increasing the same information, like just like what you call it, just not replicating it, but just yeah, just think of mitosis is being used just for growth. I don't know if you want to add anything to that.
No, that's good. Yeah.
But yeah, just make sure you have like a good understanding of this, the difference between homologous pairs and just 2N versus just N, and how these are different when you go through mitosis and meiosis. Hey guys, please ask questions. All right, so we're gonna show you a picture of a karyotype. This is a karyotype of, let me see, human, yeah. As you can see here, there are 23 pairs. So this is like one set, two set, three, third set, fourth set, and so on. So there's a total, you would call this 2N equals 46, because there's a total of 46 chromosomes, but there's 23 pairs, right? So this, if they could ask you something like, what do we call this? You would say this, we call this a karyotype of humans, right? And they'll ask you, what is a karyotype? You would say this is 2N equals 46 because it's 2N because we see pairs, and 46 because the total number of chromosomes are 46. 1, 2, 3, 4, and so on. What else can we say? So if they ask you,
versus,
yeah, so they could ask you something like, is this a sample of somatic cell or is this a sex cell? Then you would say, oh, well, I see pairs. That means there's sets, so that means that one of them is coming from mom, one of them is coming from that. So that means that this is 2N, so it's a somatic cell, right? So if they showed you just one of the chromosomes, if they just show you half of this, 1, 2, 3, 4, no homologous pairs, then you would say, okay, this is a sex cell. It's probably coming from one of the sex organs. Whereas this, you can see that this will come from, like, I don't know, this could be taken out from your liver, from pretty much any other organ.
Yeah. So, we might hold it. Sorry.
Yeah.
So, like what Jorge is trying to explain is that if you are given a karyotype and you only see, let's say for the first chromosome, for chromosome 1, you only see one chromosome instead of this pair, and you see that for the rest of them, right? Like each one for chromosome 2, there's only one, and there's only one here, and there's only one here. That means that you're dealing with the haploid cell because you don't see pairs for each chromosome set. But for this one, because you see a pair here, a pair, a pair, a pair, a pair, this is a karyotype of a somatic cell because you have pairs everywhere. And also, this is the first chromosome. It runs all the way down to chromosome number 22. Those from 1 to 22, those are all
Genetic material.
yeah, those are all from they all have they all carry genetic material. They're called autosomes.
Okay.
These are the ones that you'll see in most of the time in these are the ones that carry genetic material for your liver, for your organs, all that stuff. But for this one, the number 23, that's a special one. That's called a Germanic
Just your sex chromosome, isn't it?
sex
Sex
cells.
determined chromosome.
Yeah, these are the ones that determine your sex. So if you have an X and an X, that means you're a female. If you have an X and a Y, that makes you a male. And it's because of the certain chromosome or the certain genetic material that they carry in the X versus the Y. And so, so this is a karyotype. I'm sure you can find plenty of other pictures on the internet that are much bigger and much more in a better resolution. But yeah, this is what we're trying to get at when we're talking about the difference.
So you just remember that each pair, one of them is coming from mom, one is coming from that. And when it comes to the last pair, this right here, if you see X, this is pair number 23. If you see X and Y, and then for males, our 23rd chromosome, we have information from both our mom and our dad, whereas for females, they only have information from their mom. That's why it's only, is it XX or what is it?
No, they have information, but it's just
For the 23rd.
It's just XX, one from the dad, one from the mom.
Oh.
One X, one. Yeah. No, so the last one for the females, you're gonna get one X from your mom and one X from your dad. For males, you're gonna get a Y from your dad and an X from your mom, right? Because your mom can't give you a Y chromosome because she's a female. So you get one X from your mom, one X from your Y, or one X from your mom, one Y from your dad. And so this constitutes a karyotype. Yeah. Anything else you want to add?
I think that's it.
So, what time is it? Oof, okay. I'll just quickly, if you guys have any questions about that, make sure to add it in the comments. We're going to go really quickly through like the last bit of terminology, and then I promise I'll get through meiosis at the very end. I went through mitosis, so I want to kind of go through meiosis as well. So let me just go over some terminology really quickly, and if you have any questions, post them in the chat.
You guys okay to go? Dave, are you okay to go?
Yeah, that's fine.
You text me later. We wrap things up.
Sure, of course. Okay. Oh. Maybe I should. Oh, this is really big there. Can you tell me when it's 7:45? That way I'm not gonna go too far, too long.
You're like right there.
No, really? Yeah. Shoot, okay. Right there. So. Chromosome. So, the last thing I want to talk about is chromosomes and their terminology that's associated with them. So, when you see this right here, these two, the first two, you might think, oh, well, this is 2 chromosomes, and this is 1 chromosome. That's when chromosomes terminology gets kind of confusing. Like I said before, when we have a single chromosome and it's replicated, it's replicated, right? It's the exact same thing. So they're copies of each other. So instead, the whole thing, this right here, is considered one chromosome. However, to distinguish this one chromosome from this one chromosome that's unreplicated, we consider each one of these strands a sister chromatid. Just one in this thing. So that's a way we can kind of distinguish them. Oh, it's supposed to say sister chromatid. Sorry for the glare. But this is a sister chromatid. Sorry, this is one sister chromatid. That's the other sister chromatid. I colored them to distinguish them, but they're exactly the same copies. I only colored it so it's shown to be two different sister chromatids. Now this other one, it's still considered one chromosome. Now the thing about this that's confusing is that the way to kind of resolve this confusion is that this one chromosome has sister chromatids. This does not have sister chromatids. So this chromosome, you're going to see this before S phase, after S. Am I still in front?
You're good.
Okay. So before S phase, you'll never have sister chromatids. If you say that on the exam, I'm gonna get mad. So, but after S phase, which is the replication phase, you have this one chromosome replicates to have a clone, a sister chromatid. Now they're considered one sister chromatid, two sister chromatids. Now this last one, do you see how they're not connected at all? Well, but they're still paired up together. So what that means is that they are the same kind of chromosome, but they might come from different sources. Like we said, like we keep on saying, one is from dad. This chromosome number 1 is from dad. This other chromosome 1, that's from mom. Well, they're similar enough that they could be paired together. That pairing of them is called homologs. Those together, once they pair, those are homologs or homologous chromosomes. Now, them separate, however, the thing is they are not connected in any way like sister chromatids are. So this, you're dealing with 2 chromosomes, not 1. Even though, that is confusing. It's labeled chromosome 1, but there's 2 chromosomes to make up a homolog. Now, the thing is, about 2 chromosomes. Now, what happens when let's say this is before S phase, right? Because you don't see sister chromatids. What happens after S phase? So now these are still considered homologs, right? Because they're still the same chromosome. Now they're replicated. Now this is after S phase. Now they have, each chromosome has sister chromatids. But now, do you have 4 chromosomes or do you have 2? I wonder if anybody will answer that question. If you have an answer, put it in the comments. If you're brave enough to answer that. It's 45? Okay, just kidding. Not enough time. I'll give you the answer. It's actually just still 2 chromosomes because remember they're together, they're exactly the same. Now, if these were to split, last thing I'll say is that if these were to split, now you go from having 1 chromosome with 2 sister chromatids to 2 chromosomes. Okay? All right. That's all I want to say with the terminology. Now let's quickly go through meiosis. That way it's not false advertisement that we were going to go through both. Just wanted to get through some terminology. If you have any questions though, put them in the chat. I love the pictures in this book. All right. Can you see that? Is that small? Okay. A little bit. Yeah, it's starting to get a little hot here. Different. I'll make it squiggly. So, when it comes to meiosis 1, all right? So, because we went through mitosis, you know what, I'll move this up a little bit. All right, okay, cool. So, because we went through mitosis, you kind of understand the different stages, the different steps of each division. And so we won't go into the details of that, but what I will highlight is that for meiosis 1, you start with a diploid cell. Same thing with mitosis. The thing is, there's something that occurs in the middle of this process that changes the outcome. And that's what the big difference is between meiosis 1 and mitosis. And so the big difference is that for meiosis 1, I drew these chromosomes, right? Now, I said I start with a diploid cell. So I have 2N. So this 2N equals 2. Okay. 2N equals 2. Is that 2 equals 4? No, it's 4. Yeah, it's 4. My bad. Yeah, we just did this example. Oh, no. Okay, so does everybody see why it's 2n equals 4? The reason why is that that 2n equals 4, n would equal 2. So that means that there's 2 different chromosomes. In this case, you have this and this chromosome. So when n equals 2, that means that there's 2 chromosomes in one set. It's this very straight X and this kind of curly X. Those are the 2 chromosomes included in n equals 2. Now with 2n equals 4, that means that there's pairs. We're dealing with pairs now, which makes sense because we're dealing with diploids in this case. So now we start with this. We go through this whole phase, right? We go through prophase, we go through metaphase, we go through anaphase, and we go through telophase, eventually leading to in telophase, this cleavage furlough, where we're going to split the cells. The big difference is that when we're dealing with metaphase in this case, can you hold this real quick? Oh, you have a different marker?
Yeah.
Cool.
Is that after replication, the first bubble?
Yeah. So the big difference in this one, as you guys can tell, in metaphase, you still have that aligning of the pairs on that kind of middle of the cell, which is called the metaphase plate. But in this case, you're not splitting chromosomes like you did in mitosis. Instead, you're splitting homologous chromosomes. As you guys can tell, this right here, these are homologs, and these are homologs, correct? Because this is chromosome 1 that is similar to this other chromosome 1 from dad, mom, and these are both similar enough that they get paired together. So these are considered homologs, these are considered homologs. And so that's what you're splitting when you go into anaphase. You're splitting homologs. You're not splitting sister chromatids like you are in mitosis and eventually in meiosis 1, which I'll show you later. And so eventually, once this happens, once you're splitting homologs, so as you guys can tell, because you're splitting homologs through in anaphase, you go from this 2n equals 4 cell, which is the diploid, into these two daughter cells that are n equals 2, which makes sense because if you look, there's only one chromosome, one curly X, and there's only one X chromosome, or one straight chromosome, one straight X chromosome, and on the other one, there's also only one curly chromosome, and there's only one straight X chromosome. And so that's why you split. Once you split the homologs, you reduce from 2n equals 4 to n equals 2. Now, this is meiosis 1, and that's why, because we're splitting homologs, that's why we consider this process kind of different than mitosis. And it's because we go from 2n equals 4 to n equals 2. You don't retain the same chromosome number, which is different because in mitosis, like you guys will, like we talked about in the beginning, you retain the same number. You create identical cells that have 2n and then 2n at the end, 2n at the beginning. So now let's go through meiosis 2 really quickly. Where? Let me just summarize. What time are we at?
7:54. I don't think that's another step, it's just the products, right? You're right at the glare.
I am. Where? Ah, shit. Okay. Yeah.
Opposite way, actually.
Yeah. Yeah. Let's move it this way. Yeah. All right. So, like I said, we'll have this and this. This. So as you guys can tell, so as you guys can see from the picture, how do I do this? Okay, so as you guys can see, we go right after meiosis 1, we go right into meiosis 2. And the difference between meiosis 1 and meiosis 2 is that for this one, as you guys can tell, we have these chromosomes that have sister chromatids. And so when we go through anaphase, anaphase 2, because we're in meiosis 2, we're going to split the sister chromatids instead of the homologous pairs. And so based on that, we go from n equals 2 to daughter cells that are n equals 2, because even though this is one chromosome and that's another chromosome, like we said before, if once we split this one chromosome that has 2 sister chromatids, once they're split, they're not considered sister chromatids anymore. They're considered individual chromosomes. And so that's why we retain the 2n equals 2, n equals 2, and n equals 2, and n equals 2. The thing I didn't notice, or the thing I didn't do, was write these out or draw these out in different colors, and show you that in meiosis 1, there's a process called crossing over, in which these pairs from mom and dad, they kind of interchange material with each other. They kind of exchange genetic material, DNA, and making these chromosomes very different from or have different quantities to each other. And that's why at the very end, even though these are all in the same color and they all look the same, these all 4 individual daughter cells, they're not the same as each other. They're all different from each other because of that crossing over that occurred in meiosis 1, which is something that I would go into more, but we're running out of time. So I hope this helped. If you guys notice, last thing, if you guys notice meiosis 2, because they're splitting up sister chromatids, those are the one. That's meiosis 2 is going to be the step most resembling mitosis. And if you guys want to look at mitosis, go back in the video. We went over it pretty in depth. The steps are all the same. PMAT are all the same for meiosis and meiosis 2 as it is for mitosis. The only thing is that the end product, you're going to have 4 daughter cells after meiosis versus 2 daughter cells after mitosis. And yeah, that's about it. I hope this helped. If you guys have any last minute questions, make sure to post them. Like always, this will be posted right after this stream ends. And so if you guys want to go through it again, or if you guys want to wait until after your chem exam to go through this, feel free. This will be up, and then we are trying to do a Sunday online review. And so if you guys are interested in that, we'll post an announcement for that. It'll be online, similar to this kind of format where it's just Q&A. But yeah, thanks for joining us, guys. Make sure to sign in before you go. Yeah, we'll be here for another 2 or 3 minutes to answer any questions that you guys might have. Cool. All right, so we're going to end the stream. Thanks, guys.